Hi, it's Moser with a quick tutorial on gene linkage, or spotting genes that travel together. After you've watched this video, you should be able to explain what linked genes are and under what conditions genes can be linked. You should also be able to examine phenotype ratios in a dihybrid cross and determine if the genes in question are being passed on independently or if they're linked. And you should be able to describe, or at least name, the method used to determine if a pair of genes are linked. You already need to know how to do a dihybrid cross. Let's go. So just a quick review. You remember these things? Chromosomes? Yeah, all those pieces of DNA found in the nucleus of your cell that carry genetic information. I'll give you 2 chromosomes here for example, and you notice all those pretty bright bands on them. Now, of course, that's just an illustration. They don't really look like that, but this would be a species that has 2 pairs of chromosomes, so a haploid number of 2 and a diploid number of 4. Remember that those pretty bright bands are actually genes. Those are genes. They're literal sections of DNA that code for proteins that create some trait in an offspring. Now, in all the species that reproduce sexually, those chromosomes are arranged into pairs called homologous chromosomes. And inside each pair of homologous chromosomes is one you got from, that's right, yo mama, and one you got from yo papa. So you've got a maternal and paternal copy of each chromosome. Cool. So how can it be that you can pass on some of your mom's and some of your dad's characteristics, since the characteristics that you got from your mom are on your maternal chromosome, and the ones you got from your dad are on your paternal chromosome? Aha! Let's not forget how we make gametes. Meiosis! So during meiosis, those maternal and paternal chromosomes do a little bit of a dance. They wrap their limbs around one another and often break up, causing mixes of characteristics in offspring. We literally do some genetic mixing. No new genetic traits. This isn't a mutation. It's just a mixing of the things that you've already got on hand. Here are our little chromosomes, and they're ready to make gametes. They've been copied. They've formed centromeres. That's this thing in the middle. And now they're about to do the crossover. Now, notice that I've got a pair of genes here, gene A and gene B, and I've got a gene here, gene T, and a gene here, gene D. Keep your eye on those. Okay, they've crossed over, they've wrapped their little arms and legs around one another. That's the best I can do with this drawing technology. Now, when they separate, there have been some changes. Can you spot what they are? Note those little red lines. Above those little red lines, the ends of each chromosome have actually been torn off and exchanged. Are they the same as they were before? Nope. So, we've had some mixing and matching. The genes on chromosome 2 have been recombined. We literally call it recombination, mixing and matching. So now, instead of the dominant alleles for each gene being on the same chromosome, we have a dominant and a recessive and a dominant. But genes A and B did not recombine. Look at how close they are together. It would be almost impossible for them to get separated during the crossover phase of meiosis. We call this linked genes. When genes are on the same chromosome and very close together, they very seldom get recombined. So they tend to travel together and stay in the arrangement they are. Perhaps you've noticed that there are some characteristics in humans that seem to always go together. We see lots of redheads with freckles. We don't see a whole lot of people with black hair who have freckles. It's possible, but these are traits that might be linked. We'd have to do some experimentation to find out. We'll talk about that later. Well, how does this look in real life? Let's take an example. Let's take those T and D genes and assume we've made some gametes and had some babies. You know how to do a dihybrid Punnett square. Well, what do we get when we look at our phenotype ratios? 9 to 3 to 3 to 1. We don't even have to do the tally here. We know this. And that means that we have 56% of our offspring showing both dominant characteristics, 19% showing one dominant, one recessive, 19% showing the other recessive and the other dominant, and a little tiny fraction, 6% or so, showing both recessive genes, both recessive traits. But what about when we look at the same exact process for that A and B gene? Well, when we cross the A and B gene, and this is sort of before we know it's linked, but if we cross two organisms with those A's and B's, not knowing anything about the gametes that are formed, but only being able to see the phenotype, because remember, we can't see the genotype. What if we get something that looks like this? Whoa, 75% have both dominant characteristics. No one has a mix of dominant and recessive characteristics, and 25% of the offspring are carrying both recessive characteristics. Whoa, what's going on? Exactly. What? Remember how during the process of meiosis, the alleles for T and D got recombined? They underwent recombination because they were being sorted out to the gametes independently. We call this independent assortment. Genes A and B weren't recombined because they're linked. T and D aren't linked. T and D are far apart on the same chromosome. When genes are linked, they never, or very rarely, get separated during the process of meiosis. What does it mean if we know that genes A and B are linked? Well, it limits the number of possible combinations that we're going to see in gametes. Instead of that independent assortment where any allele from either gene can go with any other allele, where we do FOIL, the alleles are going to be distributed in the same way they're on the chromosome. So in this case, the dominant version of A will always go with the dominant version of B, and the recessive version of A will always go with the recessive version of B. This gives us only 2 gamete possibilities, which makes our dihybrid Punnett square pretty cool, down to 4 little squares. Cool, nice. If we tally up all the supposed possible combinations of genes, we get a chart that looks like this. Hey, it's 1 to 2 to 1. Yep. And our phenotype ratio is actually 3 to 1. 75% dominant-dominant, 25% recessive-recessive. Which leads us to say, how can we tell if 2 genes are linked? Well, we know what ratios we expect to see in phenotype in unlinked genes. So, looking for linked genes really just involves doing this kind of detective work. To find linked genes, scientists literally breed lots and lots and lots of animals or plants. Usually we're talking mice or guinea pigs or sweet peas, and look at the ratio of phenotype in the offspring. Dominant to dominant, dominant recessive, recessive dominant, and recessive recessive. Because when genes are not linked, also known as independently assorted, we see the 9-3 to 3 to 1 ratio of phenotypes. In other words, 56% to 19%, and 6%. But when genes are linked, that is not the ratio we see. In linked genes, we see mostly 2 of the possible combinations of genes, with none or very few of the other possible combinations. And when we see that, we start to suspect that those genes are linked. Linked genes have to be on the same chromosome, and they have to be pretty close together. The closer they are together, the fewer of those other possible combinations we will see. So, once again, how do scientists tell if genes are linked? By running repeated test crosses, breeding a whole bunch of offspring from parents who have known genotypes and looking at the phenotype ratios that show up in their offspring. Let's look at an example. For example, and this is the classic case, in sweet pea plants, flower color and pollen shape are both monogenic traits with a simple dominant recessive pattern. Purple flowers are dominant to red flowers, and long pollen grains are dominant to round pollen grains. So, when we cross a purple long homozygous dominant plant to purple long heterozygous plant to a red round heterozygous plant, we would expect to see in the offspring the 9-3 to 3 to 1 ratio. In other words, 56% of the offspring should have purple flowers with long pollen, 19% should have purple flowers with round pollen, 19% should have red flowers with long pollen, and we should see about 6% that have both recessive characteristics, red flowers and round pollen. Well, what if we breed those plants and produce 1,000 seeds and grow them and find numbers like this? 706 of them have purple flowers and long pollen, 26 have purple flowers with round pollen, 28 have red flowers with long pollen, and 240 have red flowers with long pollen. Well, what are our percentages here? 71, 3, 3, and 24. That doesn't look right. What would we start to suspect? We would start to suspect that, in fact, the genes for flower color and pollen shape are linked. Clearly they're on the same chromosome, and clearly they must be pretty close if in only 6% of the cases they end up sorted in any way other than dominant, recessive. But a gut hunch isn't enough to go on. Scientists actually use statistical analysis. That's math, folks. They use a test called the Chisquare test to tell if the difference between the expected results and the results that they're seeing, or the observed results, are due to random chance alone, or could be. So chi-squared looks like this, where chi-square is equal to the sum of, don't worry about that for now. But it's a fairly simple way to take your observed results, compare it to what you expected to get, and compare it to some known values to determine whether you should say, yeah, what we found is statistically significant, or we're not sure. It could just be random chance. Okay. Are you able to explain what linked genes are and when genes could be linked? Do you know how to examine the phenotype ratios in a dihybrid cross and tell if genes in question are being passed independently or if they could be linked? And can you describe, or at least name, the method that's used to determine if a pair of genes are linked? Excellent. Go practice.